Okay. So I would like to start this presentation with this perspective published on the New England a few days ago. So this is the State of the Union address of President Barack Obama, in which he announced that they are launching the Precision Medicine Initiative. That is a very important field, at least in oncology, because we are going to approach the genomic revolution and finally to personalize treatment according to the presence or not of specific genetic alteration in cancer. I will provide to you some examples finally. So what is precision medicine? The first message is that we can sequence the whole genome in a few days actually, and the cost is 3,000 euros. And tomorrow I am sure that in 1 hour we can do this, because the new next generation sequencing platform can sequence the whole genome in a very short time. There are some companies actually that are using this sequencing platform to provide specific indication also for our patients. This is Foundation Medicine, in which for 5,000 euro they can sequence all the genome of the tumor, of the metastatic site of disease, and finally we receive something like this, a report in which they identify specific genetic alteration that are present in the tumor, and they also indicate the phase 1 and the early phase 2 trials that are ongoing all over the world in order to potentially candidate the patient to a new treatment. So in the future, within the medical chart of the patient, we will have something like this, that is a Circos Plot. Any one of this color is a chromosome. The line between the chromosome are translocation, and the seismic line under the chromosome are the genetic alteration. That can be amplification, hotspot mutation, single mutation. You need an integration of genetic medicine and bioinformaticians in order to identify which one of this molecular alteration is the potential driver of proliferation. So you will have some genetic alteration that are drivers, some others that are passengers. But what is very important is that many clinical trials that are actually ongoing in many phase 1 units in the world are based on this. So you screen the patient with the molecular screening program, you identify a specific molecular alteration and you treat the patient according to the molecular alteration. We don't know if this genetic alteration is predictive or prognostic, but all these trials are also designed to do this. But the most interesting thing is that you can do this on a single cell with a liquid biopsy. These are exciting data presented a few years ago in San Antonio. So you just need the blood sampling and you can sequence and identify 50 different molecular alterations in the cells, in the circulating tumor cells of the patient. What we are doing in the real life, this is a trial, the SAPPHIRE-01, in which 404 patients have been included. A tumor specimen have been obtained, usually with a tumor biopsy. Then there is the molecular profiling of the patient. In this case is an exome sequencing, so you sequence just specific alteration and not the whole genome. And then you identify the molecular alteration. Finally, the bioinformatics is a statistician. You put inside the system a lot of genetic alteration, and according to statistics, you identify the mutation or the genetic alteration, that potentially is the driver. And finally, the patient has been treated within a platform of phase 1 studies according to the molecular alteration. First limitation, 404 patients enrolled in the trial, just 40 treated. Because in most of the cases, you identify alteration for which you don't have a drug, so the alteration is not druggable, or if actionable or druggable, in the center where the patient is referred, there is no trial to match with this specific alteration. So in the future, for cancer therapy, we need to identify the drivers, like HER2 or ER, and the passengers, the molecular alterations that usually are there but are not driving the cancer proliferation. So this initiative in the United States is so important that the presidents of the United States decided to announce this. And oncology is the clear choice to start a type of project like this. So in the future, at least metastatic patients will be treated according to this type of approach. So we need to design new clinical trials that are different from the prospective randomized trial, since many molecular events are very rare. So you need a global effort in order to identify 100 patients that can be eligible to an umbrella trial with a single drug. And the most interesting thing is that you will have one pathway and several diseases. So in most of the cases, the trial are not designed for a single disease, but for a single pathway and an umbrella of different diseases in which you identify the pathway alteration. And these are all the drugs under development with putative molecular alteration. And most of these drugs can induce metabolic or cardiac side effects. These are the PI3 kinase inhibitors. There are several PI3 kinase. Any company actually is developing a PI3 kinase inhibitor. Then you have AKT inhibitors with predictive factors potentially, mTOR inhibitors, the FGFR inhibitors with a spectrum of cardiovascular toxicity that is relevant. We have at least 2 phase 1 studies in my center, and this drug is really toxic for the heart. After 4 days, you have G3, G4 hypertension, very difficult to manage, and we observed also a decrease of the left ventricular ejection fraction. But it's a very promising drug because it works. We observe response. Then you have the PARP inhibitors for patients with BRCA1 or BRCA2 mutation, the BRAF mutation that is also important in other diseases other than melanoma. Then the MEK inhibitors, the c-MET, the epidermal growth factor receptor inhibitors of new generation, new HER2 inhibitors, specifically you know that some of these drugs are very active in HER2 mutations. So I am talking about new way of targeting HER2 with tyrosine kinase. Then the insulin growth factor receptor inhibitors, the MDM2 inhibitors, the CDK4/6 inhibitors that have been released by FDA a few days ago, the gamma secretase inhibitor and the Wnt inhibitors. These are the new kids on the block for which many from phase 1 to phase 3 trial are ongoing. Many of these trials will be presented in a few months in the ASCO meeting or in San Antonio for the trials in breast cancer. And for any one of these drugs, you have potential cardiovascular side effects. So what is doing the academic world and the clinical world to address this concept of precision medicine? Many cancer centers in the United States and in Europe have already a platform of genomic sequencing. In Nashville, there is one, and is dedicated to lung cancer, colorectal cancer, melanoma, and breast. Another one is the Dana-Farber Cancer Institute, then at the MD Anderson, then Michigan University, Princess Margaret Hospital in Canada, and in Europe we have UK, that they are the first to sequence the genome. If you look all the data of the whole genome sequencing of the tumors, the Atlas project, many of the PI involved are working in the Cancer Research UK, in the Sanger Institute in UK. Peter Campbell is one of the lead of this group. And they sequenced all the genome of many tumors, of different tumors. On the New England of yesterday, there is also another manuscript of this group. And then there is the Gustave Roussy and the Dutch Institute. So what is important is that many institutions in the world are investing in this new type of approach. What does it matter with cardio-oncology? Because there are similar projects also in the field of cardiovascular disease. And specifically, I mentioned this, the coronary artery disease genome-wide replication and meta-analysis, the CARDIOGRAM, in which patients are characterized with a whole genome analysis in order to identify potential drivers or risk factors for cardiovascular disease. And these are the key drivers gene that are mutated, amplified, alterated in patients with cardiovascular disease. So a similar approach from cancer medicine is moving to cardiac medicine, so to cardiovascular medicine. Look at this. These are the pathway analysis of genes specifically associated to cardiomyopathy. And if you look, many of these pathways are very common also for cancer patients. So DNA repair, inflammation, signaling of insulin growth factor, immunosignaling, vascular endothelial growth factor signaling. So there is really the concept of cross-targeting. So this is the wiring diagram of a cancer cell, and these are the most important pathway for which we are working on new drugs, and this is the wiring diagram of a cardiac cell. And I can tell you that there is an overlap of 70–80% of pathways. So much more we know about biology behind cardiac disease, much more we can prevent side effects induced by our drugs, much more we can prevent the concept of cross-targeting that Thomas Force introduced a few years ago. So what I would like to tell you is that this new approach of precision medicine will be a revolution for cancer care, but I am sure will be also a revolution for metabolic disease and for cardiac disease, because many of the concepts that we know on cells, on cancer cells, can be overlapped also on cardiac cells. So in the future we will do studies with exome sequencing, maybe on patients or in children that will develop cardiomyopathy after several years in order to understand if is there any alteration that can predict this cardiac toxicity. We can link genetic and acquired form of heart failure. We can do microarray studies, gene expression regulation studies. We can study epigenetics and also the microRNAs that are actually under investigation, like in cancer, also in the cardiac disease. What is very important, and this is my last slide, is that any type of approach like this that is very personalized, precision medicine is per definition the right drug for the right patient in the right moment. So it's so personalized that you will lose what we intend as evidence-based medicine. So in my opinion, for the future, we need to integrate this type of approach with a more scientific approach based on the analytical validity. So even if a small population, you have to be sure that this type of approach can predict the activity of a drug. We need to assess the clinical validity, so not only the prognostic value of a marker, but also the predictive value of the marker. The clinical utility, so demonstrate that what you found to be predictive of response to a new drug should improve on the outcome. We should demonstrate that this will increase progression-free survival and overall survival. And don't forget the ethical, legal, and social implication of all this testing, because while you test a whole genome analysis, you will discover a lot of new genetic information that can be related to neurological disease, can be related to genes that are associated with chronic disease, and so all these points should be addressed in order to also understand the implication for insurance, employment, privacy, and confidentiality. Thank you very much.